Good afternoon, students. Today I'm going to discuss about the reproduction in fungi. Good afternoon again to all students. So the reproduction of fungi, which I am going to discuss, and as per your syllabus, there are two fungi which needs to be discussed in detail. One of them is Mucor, and another one is an Aspergillus. So starting with today's lecture, reproduction. Okay, and most fungi basically reproduce asexually or sexually by spores. Okay, and the asexual reproduction is the most common method and produces identical organisms. And fungi reproduce sexually when the conditions are poor and nutrients are scarce Okay, so moving forward with the lecture. So fungi has various modes. So before starting the reproduction, okay, explanation for the reproduction, this figure given is just a general figure, okay, which just a general figure how fungi basically reproduces. Okay, and there are some fungi which only reproduce asexually also. Okay, in both the sexual and asexual reproduction, the fungi produce spores that disperse parent organism by either floating on the wind or hitching a ride on an animal or various other modes through which the spores can be further spread around the surroundings. Okay, so the asexual reproduction, okay, and which occurs generally by fragmentation, budding, etc. Okay, so the diagram which I'm going to discuss right now, okay, is just a generalized diagram of a fungal reproduction. Okay, and how the spores of the fungi spread, just a general idea. Okay, so mold seems to appear from nowhere, and one day apple in your food bowls are basically shiny and red, and the next day the patch of green fur appears on them. And where does the fungi come from? From where does, okay, it is comes from the spores which are present in the environments. So starts with the spores basically land. Okay, spores basically land. Okay, the air is full of mold spores, so you'll find them settling on most surfaces including the food. Okay, and fungi like mold use cells called spores. Okay, and what are the properties of the spores? They are very light and tough, so they can travel very long journeys floating in the air. Okay, and spores are like more similar to that of seeds. Okay, but they are the spores of the fungi. Okay, now what happens next? These spores grow. Okay, these spores grow. Okay, as food rods and nutrients are released, and the mold spores use these nutrients. The mold spores use these nutrients to grow. Okay, so now moving further, what will happen then? The fungus gets bigger. The fungus basically gets bigger. okay, and the mold grows into long thread-like strands, called hyphae, which we had discussed in the lecture previously. And the hyphae, okay, which contains roots, like which are rhizoids, okay, and they appear similar to the branches of the tree, okay, and the fungi takes the food through the hyphae, okay. And now moving further, what will happen next? Each sporangium contains 10,000 of spores, okay, and the basically in next stage the buds starts appearing on the tip of the hyphae. The buds start appearing on the tip of the hyphae, which are generally called sporangiospores or spores, and these are where the spores are made, okay. So now what happens at a particular period of time, okay, these birds, the round birds, which is a sporangium, basically burst, and the new spores which are present in these birds, okay, fly out, okay, and these spores are so light that they can float on the breeze, and similarly the cycle continues, okay, similarly when they move to a new environment, okay, these spores basically germinate on the given medium, okay. So what are the general properties of the spores? Spores are an adaptation to life on land, okay, and ensure that the species will disperse to the new location or basically spread to a new locations, okay. Each spores contains a reproductive cell that forms a new organism, okay, that basically forms a new organism, okay. In this case fungi will be the organism. They are non-motile in the case of fungi, okay. In the case of fungi the spores are non-motile, they may be dispersed by the wind, okay. And now moving forward with the sexual reproduction, okay, just overview, okay, I am going to go in more detail in the case of Aspergillus and Mucor. So there are also some of them which are basically dimorphic fungi, okay. When the sexual reproduction occurs, it is used when environmental conditions are poor, okay, due to the lack of nutrients, space, moisture, etc. Okay, there is no such thing which occurs in human beings, animals, etc. Okay, such as male and female pigeon. There is no such thing called male and female, but generally plus and minus signs are used to indicate different fungi, okay. And some fungi show the dimorphism, okay. We have discussed dimorphism in detail, in which fungi can grow as a single celled also, as well as the multicellular also. These are called dimorphic fungi, and they may grow as mycelia or a yeast like structure. For example, in this case filament at 25 degrees Celsius and round at about 37 degrees Celsius, okay. So now moving forward, haploid, one n hyphae from a two mating types plus and minus. As I told you, in the case of fungi we basically use plus and minus signs, not the male and female, okay, because they are not the true male and female parts, and forms a hyphae with two nuclei that becomes a zygote, okay. And the zygote divides to make a spore, okay. Zygote basically divides to make a spore, and this finally will form a spore, okay. Now the asexual reproduction, the basics one, some of the basics of the asexual reproduction. There are three types of basically asexual reproduction. Fragmentation, it is also known as vegetative reproduction also, okay. And the fragmentation is simply the part of mycelium becomes separated and begins a life on its own, okay. So if there is a fungi, okay, let us assume these are basically filaments of the fungi. Now what happens, for example, this fragment of the fungi, okay, this fragment of the fungi becomes separated, and this will grow as a new fungi. This will start growing as a new fungi. Now budding, okay, what is budding? For example, this is a cell, okay, this is a cell, okay, a small buds basically starts growing, okay, and after some time these buds get separated, okay, which we had discussed in the last lecture, get separated and can grow into a fungi, okay, and this method is basically being used by the yeasts, okay. And finally the asexual spores, and finally the asexual spores. So it occurs by the production of spores by a single mycelium, okay. Now some of the examples of the spores, different types of spores, okay. Each and every fungi has some different type of spores which are basically present in the organism. So spores may be formed directly on the hyphae, okay, inside the sporangia, or on the fruiting body. So this is a fruiting body on the sporangia, okay. You can see the sporangia here, okay, you can see the sporangia here, okay. I hope it is visible to you. And in at the tip of the hyphae, now you can see more clearly, these are basically the sporangias at the, okay. So three different fungi, three different mechanisms: Amanita, Phyllobolus, and penicillium, okay. Now moving forward with asexual, furthermore some of the more of the basics. Fruiting bodies are basically modified hyphae that make asexual spores, and this upright stalk called the spores and supports the spore case or the sporangium. Now you can see clearly in this diagram. Now see here clearly, okay, sporangiophore and sporangium, and sporangiophore basically supports the sporangium, okay. So sporangiospore basically supports the sporangium, okay. Now types of fruiting bodies. Moving forward with the types of fruiting bodies, they may be basidia, okay, given here is an basidia. Next one given here is a sporangia, it's an sporangia, okay. You can clearly see the differences between basidia and sporangia, okay. And the last ones are ascus, and also known as ascomycetes, and basidia, which represents a basidiomycetes, okay. So these structures basically help even in the identification of the fungi as well, okay. So hyphal growth, how hyphal growth occurs, okay, a germinating spore, okay, which basically grows into a mycelium and further starts growing and starts spreading around the medium, okay, and forms a mycelia. And mycelia, what is the benefit of the mycelia? It has a huge surface area, so it can absorb more amount of food, and also the larger the area, it also aids in the digestion and absorption of more amount of the food. Okay, so more important type of reproduction, as it occurs several times throughout the season and is also produced by fragmentation of the aerial hyphae, and progeny is basically genetically identical to the parent in the case of asexual, reproduction. Okay, now some more information regarding the spores. Okay, these are just the basics of the reproduction which you need to remember. The production of spore is the most common type of reproduction, and the production of spores where each spore germinate to form a germ tube that grows into a mycelium. And spores can be of various colors. It may be green, it may be yellow, it may be orange, it may be red, it may be brown, or it may be even black, and shapes may also vary. It may be oval, it may be oblong, it may be needle, helical, etc. And also depends on the number of cells, okay, which are situated at a particular arrangement, and the arrangement of cells, okay, can be easily be studied and can also be used for the identification of the fungi. Okay, so there are several types of spores, okay, which are there in different types of fungi species: conidiospores, blastospores, chlamydiospores, okay, sporangiospores, and arthrospores. And arthrospores. Okay, now moving forward. Now please have a look at the different types of spores. Okay, chlamydiospores, okay, arthrospores, blastospores, sporangiospores, and conidiospores. I'm trying to zoom the photograph image so that you can easily identify them. Okay, please have a look at the arthrospores. Okay, next one is a chlamydiospores. Okay, next ones are the sporangiospores. Okay, next one is a conidiospore. Okay, and the last one given here are the blastospores. Last one given here are the blastospores. Okay, so I'm again repeating it: blastospores, where you can see the difference between conidiospores and now sporangiospore as sac-like structures, conidiospores at the tip of the hyphae, and the terminal chlamydiospores, which contain the spores, and the arthrospores. Okay, now conidiospore, what are they? They are multiple chains or single spores formed at the sac of the end of the aerial hyphae, but they are not enclosed in a sac. Examples are Aspergillus species and Penicillium species. Examples are of the Aspergillus species and the Penicillium species. Now moving forward with the conidial fungi, it reproduces by means of asexual spores called the conidia, and conidia vary in shape and size as well as color, and through which this fungi can be identified. And most of the common household molds and the mildews are the conidial fungi. Okay, now the blastospores, okay, from where does it come from? A bud coming off from a parent cell. Of a parent cell. Okay, please have a look. Now from the parent cell, a bud will be coming out of the parent cell. Okay, now moving forward with the chlamydiospore, I did not get a very good image of the chlamydiospore, and is formed within the hyphae. Okay, for example, this one is a hyphae. Okay, this one is a hyphae. Okay, and this one is a chlamydospore. Okay, and it is a thick walled spore. It has a very thick walled spore. Okay, more of the chlamydospores. Chlamydospore is a method of producing substantial resting spore very quickly, and a nutrient is shunted from the adjacent cells into a preferred cell. It swells up and converts the nutrient materials to oil droplets for the efficient storage, and then rounds off with a thick and often roughened outer wall for the protection, where you can clearly see in the case of these spores. Okay, outer wall is basically a very thick and often roughened in structure. Okay, now sporangiospores. Okay, basically the spores which are formed within a sac, and there may be more than even 100 spores. Okay, in this sporangiospores. Example is a rhizopus as well as mucor. Rhizopus as well as mucor. Okay, now again a diagram of a sporangiospore. Okay, now this is a whole part is called a sporangium. Okay, and this the whole called is called as sporangiophore, and these ones are the sporangiospores. Okay, now you can clearly see the photograph here. It's an sporangiospore, sorry, sporangium, and which contains sporangiospore. So what is basically the sexual reproduction? Okay, what is basically the sexual reproduction? It's just the union of two nuclei, and the entire thallus can be converted into one or more reproductive structures. Okay, and the somatic and the reproductive phases do not occur together in the same individual, and thus they are called as, thus this is called as holocarpic. Thus this is called as holocarpic. So in the majority of the fungi, only a portion of the thallus give rise to the reproductive organs. Okay, so only a portion of the thallus give rise to the reproductive organs, and they are called as eucarpic. And they are called as eucarpic. Okay, and some fungal species produce distinguishable male and female organs on each thallus. Okay, but the proper signs should be used, which must be plus and minus signs. Okay, plus and minus signs, which are generally used in the case of sexual reproduction. And other species consist of male or female thalli, which may be dioecious, and therefore it cannot reproduce. Therefore it cannot reproduce by itself. Okay, and the sex organs basically are called gametangium or gametangia, which may differentiate into gametes. And the male gametangium is called antheridia or antheridium, and the female ones are called as oogonia and oogonium. But the more proper, again I told you, plus and minus signs are mostly used. And next comes the heterokaryosis. It is the existence of different kinds of nuclei. In the same individual. Okay, so there are three phases of the development in the case of a sexual reproduction. First one is the plasmogamy. Okay, it's a union of the two protoplasts, bringing about the nuclei close to together within the same cell. Okay, and the haploid nucleus of a donor cell, okay, which is generally used, plus sign is used, where penetrates the cytoplasm of the minus cell. Okay, and next comes the karyogamy. Okay, in which the fusion of the two nuclei occurs, and the formation of dikaryon a binucleate cell containing a 2 nuclei from the each parent cell and containing a 2 nuclei from the each parent cell. The 2 nuclei fuse to form a diploid nucleus. The 2 nuclei basically fuse to form a diploid nucleus. Now next third phase comes is a meiosis. Where a diploid nucleus give rise to a haploid nuclei. Where a diploid nucleus will give rise to a haploid nuclei through meiosis, and the sexual spores where some are plus or some are minus, okay, and also there are some of them are recombinants also, and these sexual spores are also used in the identification of the fungi, okay, are also used in the identification of the fungi. Now it's a diagram, okay, which contains asexual as well as sexual reproduction, okay, and this is just a generalized diagram. This is just an generalized diagram of the fungal reproduction. Okay, so basically it starts, okay, it may be in the starts with asexual one or the sexual reproduction, okay, it may start with any of the process. So let us start with this process first, okay, which is of a sexual reproduction, where haploid represents, this one is a haploid phase, diploid and a dikaryotic stage, okay. So going to start the process of the reproduction. It's just a generalized reproduction stage, okay, it's in generalized, okay, uh, a one mating type with plus and a second with a mating type minus, where plus is the donor one and second is a minus one, which is basically a recipient one, okay. Now what happens, this haploid hyphae, they are haploid, okay, or the cells of two different mating types make a contact, okay, and next these different haploid structures fuse, forming a single unit with a two genetically different haploid nuclei, forming a single unit with a two genetically different nuclei, okay. And in species that from dikaryon, the nuclei from the two mating types remain separate. They remain separate and mycelium starts to grow, and the mycelium starts to grow, and this stage is basically of a dikaryotic stage. This stage is a dikaryotic stage. The dikaryon may develop further as into the mycelium, as I told you. Okay, now comes the karyogamy. Which is basically a diploid stage, where a paired nuclei in the dikaryotic cells, fuse and further development produces, diploid zygote, okay, produces a diploid zygote. This one is a diploid zygote, and now comes the process of meiosis. And what happens, the sexual spores are basically released and producing the haploid spores of the each mating types. And what happens next, these haploid spores germinate and develop into hyphae that grow and branch to form a mycelia, So the haploid mycelium basically develops a spore producing structure. Now it may enter the asexual reproduction stage, okay, where spores of one mating type germinate and grow, okay, into new mycelium as well, or it may also enter the sexual reproduction stage, okay. Generally asexual reproduction only occurs when the conditions are good, okay, when the nutrients are easily available, and the sexual reproduction usually occurs when there is lack of nutrients, Now moving forward with the specific types, Okay, now moving forward with the specific types in the case of Mucor and the Aspergillus. So basically, just for your information, the divisions, okay, Chytridomycota, Zygomycota, Glomerulomycota, Ascomycota, and Basidiomycota. Okay, so basically fungi are divided into these divisions: Chytridomycota, Zygomycota, Glomeromycota, Ascomycota, and Basidiomycota, of which we are going to study the Zygomycota and Ascomycota. Okay, because one contains mucor and another contains a Aspergillus. Some of the examples are given here. Okay, some of the examples are given here. Basidia are generally clubbed fungi. Zygomycota are generally bread molds. Chytridomycota are generally chytrids. M fungi are also known as arbuscular mycorrhizal fungi, which are basically mycorrhizas. Ascomycota are basically sect fungi, and lichens are basically association between algae and fungi. Okay, now this one is a diagram of a generalized zygomycota, and zygomycota basically has a sporangiophore. Okay, basically has a sporangiophore, and it contains sporangiospores. Okay, for the reproduction. Now please have a look of the various uh, fungi of the group Zygomycota rhizopus. Okay, okay, and the hyphae do not have cross walls, and the hyphae do not have the cross walls. They grow rapidly and also includes Rhizopus stolonifer, which grows in the bed, and the asexual reproductive structure called sporangium. Okay, called sporangium. A sporangiospores make the spores, and the rhizoids given in the diagram anchor the mold and release the digestive enzymes and absorb the food, and stolons connect the fruiting bodies, and stolons connect the fruiting bodies. Okay, now zygomycota, yes, we have studied, some of them produce zygospores. Okay, the sexual spores are produced by the conjugation, where plus hyphae and minus hyphae, where one is donor and another one is a recipient hyphae. The sexual spores are called as zygospores, and zygospores can endure harsh environments until the conditions improve. Okay, this one is a photograph of a zygospore. It's a zygospore. Okay, now mucor, one of the organism which we are going to discuss today. Okay, we have just completed our basics of the reproduction, so I will be not explaining the each and basics now. Now directly moving with the mucor. This one is our photographs of the mucor, electron microscope, light microscope, and the growth on a agar medium. Okay, and mucor reproduces by vegetative and sexual means, and the vegetative reproduction, okay, which occurs by fragmentation, due to the accidental breakage, and the mycelium may break up into two or more units, and each unit is capable to grow as a mother mycelium. So fragmentation we have already discussed, where a fragment, okay, a part can grow on its own. Now the second one is a sporangiospore. The asexual mode is a sporangiospore. During favorable conditions, the non-motile spores known as sporangiospores or aplanospores are formed inside the sporangium, and the sporangiophores develop singly or scatteredly on the upper side of the artificial mycelium. Okay, which we already seen the diagram. Now next comes the odia. This one is the first one. What are odia? Basically they are thin walled bead like structures formed by mycelium, okay, and grow in a medium which are rich in sugar, and after the detachment the odia increase by budding like a yeast, and this stage is called a torula stage, which may later develop into a mycelia. So these structures grow in a medium which are basically rich in sugars. Okay, they produce bead-like structures which can grow like a yeast, okay, like a budding, and this stage is called a torula stage, and which may later on develop as an mycelial stage. Now comes the chlamydiospores. We all know the property of the chlamydiospores, and in the case of Mucor, during the unfavorable conditions, thick walled nutrition rich intercalary mycelial segments are developed by the separation of the mycelium, which are termed as chlamydiospores. In the favorable conditions, the chlamydiospore germinates and gives rise to a new mycelium and gives rise to a new mycelium. Okay, please have a look at the diagram given here. Okay, please have a look at the diagram given here. Okay, this is just a cyclic process. Okay, where a vegetative germination occurs. A vegetative mycelium which may develop chlamydiospores, okay, or it may also produce sporangiospores, okay, depending on the condition. Okay, this is just a cyclic process how these spores are formed. Okay, vegetative mycelium, a prostrate hyphae, young sporangiospores. Okay, a swollen hyphal tip develops. A swollen hyphal tip will be further converted into a larger one, okay, which contains a sporangium, and further after the development of the sporangiospores, after some time, these sacs, okay, due to the spores burst out, and the sporangiospores are set free, and these sporangiospores can germinate again, and similarly chlamydiospores are formed in the case of mucor when the nutrients are scarce. Okay, now moving forward with the reproduction, where I have also included a sexual reproduction stage. You can see it is almost similar. It is also almost similar to the generalized one, but in this case zygospores will be formed. Okay, please have a look at the diagram. Next, all of them, all the parameters are the same. Mating occurs, haploid stage, gametangia, okay, and plasmogamy occurs, okay, and the cell wall thickens and produce a dikaryotic zygospore develops, okay, and the karyogamy occurs where the both the nuclei fuse, and finally a meiosis occurs, and the new mycelia will be formed from the germinating spores. Okay, I'm repeating again, starting with this one here. The hyphae of the two mating strains, donor and recipient, make a contact, and the septum forms behind each hyphal tip, isolating a haploid nuclei into gametangia. Okay, and you can see next the gametangia. This gametangia fuse and plasmogamy takes place. In the third point, the cell wall thickens as a dikaryotic zygospore develops. And the fourth point, the karyogamy occurs where both nuclei pair and fuse, forming a diploid nuclei. Okay, which produces a single multinucleate zygospore or a zygote. Okay, and after months or years, when the conditions are favorable, okay, and zygospores basically germinates and splits open, producing a sporangium. producing a sporangium which produces haploid spores through the meiosis process, and each spore will develop into a new fungi. Okay. Now same it's just for your diagram. You can draw any of the diagram in this case. Okay, you can draw any of the diagram whichever you want to draw. Okay, so now moving forward with the ascomycota. With the ascomycota group, what are the characteristics of the ascomycota? They are sac fungi. They are sac fungi, which includes scab fungi. Why are they known as sac fungi? Because they produce a sac-like structure for the spores. Morels, next examples are truffles, yeast, and mildews. Okay. They may be plant parasites also in the such as Dutch elm disease and chestnut blight, and they reproduce sexually as well as asexually, and their specialty is the production of ascus. Okay, basically sex that makes ascospores, a sec that makes ascospores in the sexual reproduction. And the specialized hyphae known as ascocarps contains asci, and the specialized hyphae known as ascocarps basically contains asci. Okay. Now furthermore characteristics: yeast reproduce sexually by budding, and asexual spores are called conidia and form at the tips of the special hyphae called conidiospores. Okay, this one are the conidia. Okay, conidia formation. Okay, next is a conidia formation. You can see the diagram here. Okay, now moving further with the aspergillus. Please have a look at the photograph of the aspergillus. Photograph of the aspergillus. Now starting with the aspergillus, they reproduce by vegetative, okay, asexual and the sexual methods. It takes place by fragmentation in the case of vegetative and the sclerotia. Okay, fragmentation we have already studied. What do you mean by fragmentation? The mechanism is the same and grows under the favorable conditions. Sclerotia. Next comes the sclerotia. Some species, example Aspergillus niger, Aspergillus terrus, produce sclerotia. It is more a means of keeping fungus alive than the propagation. It's just for the survival of a Aspergillus for a longer time. Okay. Next comes the asexual reproduction, which takes place by the hyphae, called conidiospores, or spores, and some cells of the fungi are known as food cells also, and each food cells produce a special erect branch as an outgrowth, and it is the young conidiophore. The tip of conidiophore develops into an elliptical or a globular multinucleate head called vesicle, where it forms a radially arranged tubular growths called sterigmata and or phyllids. Okay, and some species primary sterigmata or unicellular bear secondary sterigmata also. Okay, please have a look given here at the diagram given here. Okay, in this case, if a asexual reproduction, where a young conidiophore arising from a foot cell, okay, and B from diagrams B to E, okay, is the formation of primary stigmata. Now please have a zoomed look here. Okay, a foot cell in the diagram A given here. Okay, now what will happen in the diagram B? If from the foot cell a coloniophore will be formed and a young vesicle also. And in the diagram C, you can clearly see the sex, which will start forming the sex. Okay, and the D, the primary sterigmata will be formed. The primary sterigmata will be formed. Okay, now in this diagram, and in this diagram, where you can see A, in the case of a asexual reproduction, okay, where a mature conidiophore with the primary stigmata, and the diagram B, okay, with a secondary stigmata, okay. Now comes the third one, how the conidia are formed. Okay, it starts with a young phyllid, okay, and you can see the division here of the nuclei, and the conidium initial, which you can see where a new wall will be formed. Okay, and the first conidium basically is formed. First conidium in this case basically will be formed, as at the same time new second conidium will be also formed, and in the next, similarly, it grows on and grows on, developing a 3, 4 depending on the species of the fungi. Similarly, different number of conidium will be formed. Okay, now again, germination of conidium. How will this conidium germinate? Okay, a conidium which will be released and starts to germinate. Okay, similar to that of the other fungi as well. Okay, now comes the sexual reproduction. You can draw a diagram whichever you want. It will primarily depend on you. The mechanism is same. Okay, mechanism same to that of the other fungi, but in this case, ascospores or the asci, because this one is an sec fungi. Okay, starting with the first point, hyphae of one mating type fuse to hyphae of the opposite type. Okay, where a plasmogamy occurs and producing a dikaryotic ascus. And next comes the karyogamy, where both the nuclei fuse, producing a diploid zygote. And now from the meiosis, in the diploid nucleus produces a 4 haploid nuclei. Okay, and the 4 nuclei basically, which is seen here, will now divide by the mitosis, and then the cell walls form from each of the resulting 8 nuclei, and these cells are basically ascospores. Okay, the asci develop within a ascocarp, and which began to form soon after the sexual reproduction. And finally, the asci release their ascospores from the opening of the ascocarp, and they may move forward for the asexual or the sexual reproduction, depends on the environmental conditions of the surroundings. Okay, so now this one is just a basic diagram of the sexual reproduction diagram of the Aspergillus. Okay, it is almost same. Whichever diagram you want to draw in the diagram, it's on you. Purely on you. You can draw any of the diagram whichever you want.